Hi everybody, this is Mr. Yeoman, and today we're taking a look at mutations. Mutations are a very important part of the way that DNA functions. It is the raw material for evolution. In order for organisms to change, they have to mutate, they have to get changes to that DNA. Now this can happen in one of two ways. This can happen because of a replication error. The DNA polymerase goes through, and as it's putting in the nucleotide bases, then it might just put in the wrong base by accident. Most of the time, the DNA polymerase is going to go back and proofread that new strand and fix its mistake. But every once in a while, one's going to slip through, and we end up with a mutation in our DNA in that cell. The other type is mutagens. Now, these mutagens can come in a variety of ways. The most common one is radiation, for example, and so there are lots of different types of radiation, but they're all going to end up damaging that DNA. And so it might not be very strong radiation, that it only damages cells on the surface, or it might be really, really strong DNA radiation, like gamma radiation, that is going through and just breaking that DNA apart deep inside of you. But there are others as well. So chemicals, we know that certain types of chemicals, whenever we burn something, it ends up acting as a mutagen and changing the DNA. And then viruses, one that I've talked about in class several times is HPV, human papillomavirus. We know that it is the primary cause for cervical cancer. So there are all these different types of mutagens that can end up changing our DNA, and that can have a variety of outcomes. So the simplest and the one that's going to apply the most to this particular unit is nucleotide mutations. So this is where you have a single nitrogenous base or nucleotide that is going to change in some way. So I like to represent this with a simple phrase: the fat cat ate the big rat. I like to use this phrase because all of the words within the sentence are three-letter words, just like the codons within RNA. Some of the different outcomes for this is you could have a substitution. So in this first case, we could have take the F in fat and substitute in an S. And so now we end up with the fat cat ate the big rat. We did change something, but overall, the majority of our sentence is pretty much the same. Another way that we could change this is the fat cat ate the big rat by substituting in a K instead of a C. Now, when I read it, it's still the fat cat ate the big rat. Okay, this I would refer to as a silent mutation because it really didn't change anything. Okay, we did change the letters, but the end result is it ends up coding the same way. And we see this because of that redundancy that we find in the codon chart, is that we have multiple codons that will code for the exact same amino acid. So we might end up having a mutation that still results in the exact same protein amino acid sequence. That's different than the last one because that would be a missense mutation. We are changing part of it. We're changing one letter, which does slightly change the meaning of the sentence, but it might not be enough to completely change it. We might still end up having a functioning sentence, or in reality, a functioning protein molecule. The last one I have here is just the fat cat. This is when we get a substitution that creates a premature stop codon. So we call this a nonsense mutation because our sentence just stopped and we're just left with nonsense. The fat cat did what? We don't know because it stopped too early. And so this is not going to result in a functioning protein. Those are the three that we end up seeing the most when we're looking at functioning biology, because the next ones, the insertions or deletions, which I most often refer to as indels, these indels are going to end up causing a big problem. In this case, I'm going to insert an L between the C and A of the cat. And so when I do that, this is going to result in a frame shift. And so that moves down all those nucleotides. And so now the order, the sets of 3 that previously existed, no longer exist. Now they're coding for something else. The difference is now that we see gibberish in the sentence that I made, when we're looking at the messenger RNA, it's not going to be gibberish. They do still code for something. It's just coding for something totally different. The same thing will happen with a deletion. So in that next line, what I did is I deleted the A out of fat. And so that resulted in a different frame shift. So all of those letters ended up moving back down and changing that reading frame. And so now, again, it looks like gibberish to us, but keep in mind, this wouldn't be gibberish to the ribosome. It's still going to be coding amino acids, but they're going to be totally different amino acids than the sentence that we started with or from that messenger RNA strand that we began with. So those are the 3 types of nucleotide mutations that we're going to be taking a look at, and it's going to be the majority of the things that we are discussing within this unit. That doesn't mean there aren't other types of mutations, and so I want to briefly talk about these other types, even though we're not going to be looking at them in particular in this unit. So rather than a nucleotide mutation, we could have an entire gene mutation. And so in general, these will keep the entire genes intact, but it will just like move them around. And so taking a look at this, I've drawn a chromosome with genes A, B, C, and D. So this is just a made-up chromosome with some random genes on it. One of the first things that can happen is create a duplication. So you can actually make a section of your chromosome where there's multiple genes on it, or maybe even just a single gene, but you're copying this whole section. So we get another section of genes on that chromosome. This can be beneficial because we can code multiple times for that same gene, or it might allow us to mutate, have those single nucleotide mutations within that spare gene without getting rid of the original. And so sometimes we'll see gene families within our DNA, and a lot of times those have originated as the same as just duplications of that same gene. The next option is a deletion. So we are going to remove, in this case, we removed the B gene from it. Again, depending on what that gene is, it might cause huge problems. It might not cause that many problems at all. But it is just deleting that entire gene out, or at least enough of it that that gene no longer functions. Another example is an inversion. So now the genes are still going to be present, they're just in a different order. This makes a bigger difference when we start taking a look at things like crossing over. So the closer the genes are to the end, the more likely they are to cross over during cell division, during meiosis. So that can end up playing a slight role in it, but the genes are all still present. To me, one of the most interesting examples of gene-wide mutations is translocation. And so what I'm drawing here is that I have that original chromosome with A, B, C, and D on it, but I'm also going to have a second chromosome, which have genes E and F on it. In an instance where we have translocation, what that means is that we're moving the gene from one particular chromosome onto a different chromosome. There are lots of examples of this, but I think that one of the most interesting examples of it is looking at the Y chromosome. Many people think of the Y chromosome as being the defining characteristic of what makes a human biologically male. But to be more specific, it's the SRY gene that is typically located on the Y chromosome. Now, what has happened in some instances is that SRY gene, which then turns on a bunch of other genes which provide all of the secondary sex characteristics that we typically associate with being male, that ends up getting moved onto another chromosome. And so this has happened with some individuals where that SRY gene has been put onto a different chromosome, particularly the X chromosome. So there are some individuals that have two X chromosomes, so they would be chromosomally female. But because the SRY gene is present, it turns on all of these other genes, which then they end up appearing they are biologically male because they have that SRY gene. So I think that's just an interesting example. And the opposite is true. If the SRY gene has been taken off of the Y chromosome, then that means that even if you have the Y chromosome, it's not going to turn on all of these other genes that are typically associated with being male. And so I think, again, one of the really, really interesting cases of this, there's a woman in the UK who, through a series of other genetic tests related to cancer, she found out that she had a Y chromosome, but there was no SRY gene on that Y chromosome. And so her remaining X chromosome she is biologically completely female, and to the point of she has 2 children of her own. And so she is a fully functioning female, but has a Y chromosome. So I think that's another interesting just example of how this translocation can work. The last type of mutation that I want to address is a whole chromosome mutation. And so a lot of times we'll refer to this as aneuploidy. It's just having an atypical number of chromosomes. And this most often happens during meiosis, when the chromosomes are dividing, and instead of dividing the way that they're supposed to, where they are going to separate out and go into 2 separate cells, instead they stay stuck together, and so you end up with an extra chromosome in one cell and not enough chromosomes in the other one. And so there are some different examples of that, like Down syndrome is probably the most notable example, but there are some others that we'll talk about in our next unit. Turner syndrome and Klinefelter syndrome are another couple of examples of chromosomal mutations. So I hope that helped. And as always, stay curious and seek truth.